This video demonstrates a method in which UV light is used to manipulate the activity of a protein at the plasma membrane. To do this, cells are co-transfected with membrane-anchored FRB protein and the FKBP-fused protein of interest. A photocaged rapamycin derivative is added to the medium. Then, during live cell imaging, UV light is applied to a specific region of the cell. The linker between rapamycin and biotin is cleaved, releasing chemical dimerizer and its byproduct. The released dimerizer diffuses into cells and induces dimerization between the FKBP-labeled protein of interest and plasma membrane-anchored FRB in the irradiated region. Without the UV radiation, FKBP and FRB do not associate, hence producing no effect. Analysis of data reveals highly localized recruitment of the FKBP-fused protein of interest based on its physiological effect.
The advantage of this technique over existing methods such as pharmacology and genetics approach is the combination of speed of action, target specificity, and spatial resolution. The implications of this technique extend toward therapy of cancer. Metastasis is a consequence of migration by cancer cells. Therefore, manipulation of activity of key migration factors may reveal its precise molecular mechanism.